Number 10. Pithovirus sibericum, the giant virus that woke from the ice in 2014. A team of French researchers announced a discovery that changed how scientists think about the frozen past. From Siberian permafrost, they recovered a virus that had been buried for roughly 30,000 years. That means it had entered the ground during the Pleistocene, when mammoths still crossed the tundra, when Neanderthals still existed, and when human civilization was not even a distant possibility. The virus should have been dead. Its genetic material should have broken down. Its structure should have collapsed. Instead, when scientists thawed it in the laboratory, it began to function again. They named it Pithovirus sibericum. Pithovirus is not an ordinary virus in the way most people imagine one. It belongs to a strange category called giant viruses, organisms so large and genetically complex that they blur the boundary between textbook definitions. Some giant viruses are big enough to be seen with a standard light microscope. They can carry hundreds of genes, more than some simple bacteria. Pithovirus looked less like a tiny invisible particle, and more like a biological machine from a forgotten world. But here is where the discovery becomes disturbing. Pithovirus did not merely preserve ancient DNA. It remained infectious. When researchers exposed it to amoebae, it invaded the living cells and began replicating, as if the 30,000 years under the ice had been nothing more than a pause. From the virus's perspective, no empire had risen, no language had been invented, no species had gone extinct. The world had changed completely, but the virus had not. It had simply waited for warmth and a host. That is the true horror of Pithovirus. It proved that permafrost is not always a graveyard. It can be a freezer. For decades, many scientists believed that viruses could not remain viable across such vast stretches of time. Cold might slow decay, but eventually radiation, chemical damage, and molecular breakdown should make infection impossible. Pithovirus showed that the rule was not absolute. Under the right frozen conditions, a virus can remain intact for tens of thousands of years. The reassuring detail is that Pithovirus infects amoebae, not humans. It was not a revived human plague. It did not leap from the laboratory into people, but that detail is only partly comforting. Scientists used amoebae because they are standard hosts for studying these giant viruses. The point is not that Pithovirus itself will cause the next pandemic. The point is that it proved ancient viruses can come back infectious. If an amoeba virus can survive that long, then the question becomes unavoidable. What else can? Siberian permafrost contains the remains of animals, plants, microbes, parasites, and entire ecosystems that were buried during the Ice Age. Mammoths, woolly rhinos, horses, reindeer, wolves, and human ancestors all carried viruses when they died. Some of those viruses died with them, but Pithovirus tells us that some may not have. The ice does not care whether a pathogen is harmless or dangerous. It preserves whatever conditions allow it to preserve. And now those conditions are changing. Arctic regions are warming rapidly. Permafrost that stayed frozen for tens of thousands of years is beginning to thaw. Soil collapses. Riverbanks expose old layers. Ancient organic material enters meltwater. Animal carcasses emerge from the ground with skin, hair, organs, and microbes still preserved. The same kind of frozen world that held Pithovirus in suspension is no longer stable. That is why Pithovirus sibericum belongs among the prehistoric plagues scientists fear could return. It was not dangerous because of what it infected. It was dangerous because of what it proved. It showed that deep time does not always destroy biology. It showed that a virus older than agriculture, older than cities, older than recorded memory, could still wake up when the ice was removed. The most unsettling part is that Pithovirus was discovered because scientists looked. Countless other frozen samples have not been tested. Countless ancient soils have not been cultured. Countless buried organisms are still locked beneath the Arctic surface. The first revived giant virus was not necessarily rare. It was simply the first warning to come back alive. Pithovirus did not return as a fossil. It returned as a functioning pathogen in its own small world, and that changes the meaning of every thawing layer of permafrost. Beneath the ice, the ancient world may not be finished. It may only be waiting for the temperature to rise. Number 9. Mollivirus sibericum. The second sleeper from the same frozen world. One ancient virus could be dismissed as a freak accident. a biological miracle, a rare survivor preserved by perfect conditions in one patch of Siberian soil. That is what made Pithovirus terrifying, but still possible to explain away. Maybe it was exceptional. Maybe science had found the one impossible virus lucky enough to survive 30,000 years in the ice. Then, 1 year later, researchers returned to the same frozen world and found another one. In 2015, the same French research team announced the revival of Mollivirus sibericum, another giant virus extracted from Siberian permafrost, around 30,000 years old. It came from the same deep time as Pithovirus, the same Pleistocene world of mammoths, frozen soil, and vanished ecosystems. But it was not the same virus. It did not simply belong to the same family. It was structurally different, genetically different, and biologically distinct. That detail changed everything. Pithovirus had shown that one ancient virus could survive. Mollivirus showed that the ice could preserve more than one kind. This was no longer a single biological anomaly, it was a pattern. Two different viral lineages, buried for tens of thousands of years, had remained capable of infection after thawing. The permafrost was not holding one lucky survivor. It was holding diversity. Like Pithovirus, Mollivirus infects amoebae, not humans. In the laboratory, researchers exposed the revived virus to living amoeba cells, and the virus began replicating. It did what viruses do. It entered a host, used the cell, and made copies of itself. The frozen time between the Ice Age and the modern laboratory had not erased its function. It had only paused it. From the virus's point of view, the 30,000 years in the ground were not history. They were storage. That is the disturbing lesson of Mollivirus. Permafrost can preserve entire viral families, not just isolated particles. A single soil sample can contain more than one ancient infectious agent. If two different giant viruses could be revived from a tiny fraction of the frozen Arctic, then the total number of dormant viral forms hidden across permafrost must be far larger than anyone can test directly. The Arctic is not one freezer box. It is a continental archive. The scale matters. Siberian and Arctic permafrost stretches across millions of square kilometers. It contains layers of soil, ice, plant matter, animal remains, microbes, and ancient organic material built up across thousands and sometimes hundreds of thousands of years. Every buried organism carried its own microbial world. Every carcass, every root, every layer of frozen mud may preserve genetic material from ecosystems that no longer exist. Mollivirus tells us that some of that material may not be dead. The reassuring part again is host range. Mollivirus was revived using amoebae, and there is no evidence that it threatens humans. But the method of discovery creates a blind spot. Scientists revive amoeba viruses because amoebae are useful and safe hosts in this kind of work. That means amoeba viruses are the ones most likely to be found first. It does not mean they are the only ancient viruses preserved. It means they are the ones we have been looking for. The darker question is what remains untested. What about viruses that infected mammals? What about viruses adapted to Ice Age horses, reindeer, wolves, cave lions, mammoths, or early humans? What about ancient viruses whose modern hosts are gone, but whose biology might still recognize something close enough in modern animals? Mollivirus does not answer those questions. It makes them impossible to ignore. And now the environment that preserved these viruses is changing. Arctic warming is no longer a future scenario in scientific reports. It is already reshaping the ground. Permafrost is thawing. Ice, rich soil is collapsing. Ancient layers are being exposed to air and water. What laboratories do carefully with selected samples, the climate is beginning to do across entire landscapes. It is thawing the archive without reading the warning label. That is why Mollivirus sibericum belongs among the prehistoric plague scientists fear could return. It was not frightening because it was known to infect humans. It was frightening because it proved Pithovirus was not alone. The first revived giant virus was a warning. The second was confirmation. Deep inside the frozen soil, ancient viruses were not waiting as fossils. They were waiting as possible functions. Mollivirus changed the question from can one virus survive deep time to how many have survived. And that is where the fear becomes larger than any single organism. Because the permafrost is not revealing one monster. It may be opening a library of them, written in genomes no living immune system has ever had to read. Number 8. Pandoravirus yedoma. The ancient virus with no modern map in 2023. Researchers studying Siberian permafrost pushed the warning deeper into time. After Pithovirus and Mollivirus had already shown that 30,000-year-old viruses could remain infectious, scientists revived another giant virus from frozen ground. This one was older, much older. It came from yedoma permafrost, a type of ice, rich ancient soil known for preserving organic material from the Pleistocene. The virus was dated to roughly 48,500 years old. They named it Pandoravirus yedoma. The name was not accidental. Pandoraviruses were named after Pandora's box, the mythic container that released suffering into the world when opened. These viruses are enormous by viral standards, with giant genomes and genetic features that do not fit neatly into familiar categories. Many of their genes have no clear match in modern databases. That means when scientists sequence them, they are not simply looking at an old version of something they already understand. They are looking at biological information with large sections that remain unknown. That is what makes Pandoravirus yedoma more unsettling than age alone. A virus can be old and still familiar. This one is old and strange. Its genome carries a large amount of genetic material that does not easily compare to known organisms. Modern medicine depends heavily on comparison. When a new pathogen appears, scientists look for relatives. They compare sequences. They search for known proteins, known vulnerabilities, known mechanisms of infection, known patterns that suggest how to detect it or stop it. But an ancient virus with many unfamiliar genes does not offer that comfort. Like the earlier revived giant viruses, Pandoravirus yedoma was tested on amoebae, not humans. It is not known as a human pathogen. But again, the danger is not only the individual virus, the danger is the principle it proves. If a virus can remain infectious for nearly 50,000 years, then the timeline of biological survival in ice is far longer than many people assumed. The frozen past is not limited to a few centuries. It is not limited to the age of written history. It reaches back into a world before agriculture, before cities, before the extinction of the last Neanderthals, before the cave art that later humans would leave behind. From the virus's perspective, none of that history matters. It did not experience 48,500 years as time. It experienced cold suspension. When the conditions changed, when the sample was thawed and a compatible host appeared, the virus resumed what it was built to do. Replication did not need memory. It needed opportunity. This is where permafrost becomes terrifying. The deeper scientists look, the older the revived viruses become, first 30,000 years, then nearly 50,000. But yedoma deposits can preserve material even older than that. The limit has not truly been reached. We do not know the maximum age at which a virus can remain infectious in frozen ground. We only know the oldest one we have successfully revived so far. And the Arctic is now creating accidental experiments across enormous landscapes. As temperatures rise, yedoma permafrost thaws, frozen cliffs collapse into rivers. Ice, rich soil turns into mud. Ancient layers that were sealed since the Pleistocene are exposed to air, water, animals, and people. A laboratory revival happens under containment. A landscape thaw does not. It releases whatever is present into the world without selection, warning, or control. Pandoravirus yedoma also raises a deeper scientific fear: recognition. A pathogen with familiar genetic signatures can be detected more quickly. A strange one may not be recognized at first. It may appear as noise, contamination, an unknown syndrome, or a mystery infection. Modern surveillance systems are powerful, but they are built around what we know how to look for. Ancient biology may not follow modern expectations. That does not mean Pandoravirus yedoma itself will infect humans. It most likely will not. But it proves that the ice can preserve giant viruses from a time so distant that their genetic world feels almost alien. If harmless amoeba viruses from that age can survive, then ancient viruses of unknown host range may also survive somewhere in the same frozen archive. That is why Pandoravirus yedoma belongs among the prehistoric plagues scientists fear could return. It is not frightening because it has already caused disease in humans. It is frightening because it expands the scale of the problem. The permafrost is not only preserving old pathogens, it is preserving old unknowns. A virus can wait nearly 50,000 years and still be biologically active. Its genes can be strange enough that modern science does not fully understand what they do, and as the ice opens, the question is no longer whether ancient viruses can wake up. The question is how many we will recognize before they do. Number 7. The 1918 influenza, the pandemic preserved in Arctic graves in 1997. A pathologist named Johann Hultin traveled to the village of Brevig Mission, Alaska, to dig into one of the darkest graves of the 20th century. In November 1918, the influenza pandemic had struck the village with devastating speed. In only a few days, most of the community was dead. The victims were buried in a mass grave in permafrost, sealed by the cold in a place where decomposition slowed, and the bodies remained preserved long after the world moved on. Hultin was not looking for bones. He was looking for a virus. The 1918 influenza pandemic had killed tens of millions of people around the world, perhaps more than the First World War itself. But for decades, scientists did not fully understand what made that virus so lethal. It had moved through the human population with terrifying force, killing not only the very young and the old, but also healthy adults in the prime of life. To study it properly, researchers needed preserved tissue, and in Brevig Mission, the frozen ground had done what no archive could do. It had kept pieces of the pandemic physically intact. From the preserved lung tissue of one victim, researchers were eventually able to recover viral genetic material. By 2005, scientists had reconstructed the 1918 influenza virus in a high-containment laboratory. The pandemic was no longer only a historical event described in death records and old newspapers. Its genome had been sequenced. Its biology could be studied. In a controlled scientific sense, one of the deadliest viruses in human history had been brought back into the modern world. That achievement was a medical milestone, but it also carried a warning. It proved that viral blueprints can survive inside frozen human remains for nearly a century, intact enough for science to reconstruct a pathogen that once reshaped the planet. The Brevig Mission grave was not magic, it was permafrost. And if permafrost preserved 1918 influenza there, then other frozen graves across the Arctic may preserve other deadly pathogens in the same way. This is where the horror becomes larger than one laboratory. The 1918 pandemic did not kill people only in Alaska. It reached Russia, Canada, Norway, Greenland, Siberia, and countless cold regions where victims may have been buried in frozen ground. Some of those burial sites are now being exposed by erosion, construction, flooding, and thawing permafrost. A grave that was once sealed by climate can become unstable when the ground warms. What was buried as history can return as biology. The 1918 virus is also frightening because influenza is not a static enemy. It mutates, it reassorts, it crosses species barriers, it can move between birds, pigs, humans, and other hosts under the right conditions. The reconstructed 1918 strain showed features that helped explain its unusual virulence. But it also reminded scientists how dangerous influenza can become when a population has little protection against it. Modern medicine is far stronger than it was in 1918. We have antivirals, hospitals, vaccines, genome sequencing, and global surveillance. But those systems are not perfect. A resurrected or released historical strain would enter a world where most people have no direct immune memory of the original virus. The 1918 pandemic is old enough to be history, but recent enough that its grave still exists. It sits in an unsettling middle ground, not prehistoric, but preserved by the same frozen systems that hold far older threats. The most disturbing part is that the virus did not need to survive as an actively infectious particle in the grave to change the future. Its genetic fragments were enough. Once science has the blueprint, biology can be rebuilt. That is useful for understanding disease and preparing defenses, but it also proves that frozen bodies are not only burial sites, they can be libraries of extinct or vanished pathogens, storing instructions that modern technology may be able to read. That is why the 1918 influenza belongs among the prehistoric plagues scientists fear could return. It shows that frozen human remains can preserve the memory of pandemic disease in a form science can recover. It shows that permafrost graves are not always sealed endings, and it shows that the past does not have to walk out of the ice alive to become dangerous. Sometimes all it needs to leave behind is a code. The 1918 pandemic killed faster than many societies could bury their dead. A century later, one frozen lung helped bring its virus back into scientific existence. The grave at Breivik mission was opened by human hands carefully and deliberately. But across the Arctic, the ground is opening on its own. And if the ice still holds old pandemics, the question is not only what killed the dead, it is what the dead may still be carrying. Number 6. Anthrax. The prehistoric killer that has already returned in the summer of 2016. The Yamal Peninsula in northern Siberia was hit by a heat wave that felt wrong for the Arctic. Temperatures rose far above normal, the tundra softened, permafrost that had held the ground stable for generations began to thaw deeper than usual, and from that thawing ground, something emerged that had been buried for decades, the frozen remains of a reindeer that had died during an anthrax outbreak. The animal had been dead since the early 20th century, but the disease inside it had not vanished. Anthrax is caused by the bacterium Bacillus anthracis, and it has one trait that makes it especially terrifying in frozen landscapes. When conditions become hostile, it forms spores. These spores are survival capsules, hardened biological structures that can endure cold, heat, dryness, chemicals, and time with extraordinary resistance. They do not need to grow. They do not need to feed. They simply wait. When the reindeer carcass thawed, the anthrax spores were released back into the environment. The result was not theoretical. It was not a laboratory warning. It was an outbreak. More than 2,000 reindeer died. Dozens of people were hospitalized. A young boy died after exposure through contaminated meat. Russian authorities declared an emergency, sent biological response teams, and worked to contain the spread before it moved farther through herds and communities. This is why anthrax belongs near the center of this list. It is the proof that ancient or long-buried pathogens do not have to wait for science to revive them. The planet can do it by itself. A warm summer, a thawed carcass, grazing animals, human contact, and a spore built to survive. That was enough. The outbreak did not escape from a lab. It came out of the ground. The deeper horror is that the Yamal event may not be unique. Across Siberia and other cold regions, animals have been dying of anthrax for centuries. Their bodies were often buried in frozen soil or left in ground that stayed cold enough to preserve them. Reindeer, cattle, horses, and other livestock carried spores into the permafrost with them. Some burial sites were marked, many were not. Others have been lost under snow, mud, and time. As the Arctic warms, those old burial places become unstable. Ground collapses, riverbanks erode, soil layers open. What was once locked beneath the tundra can enter grass, water, insects, animals, and people. Anthrax does not need a complex chain of modern adaptation. It already knows how to infect mammals. It already exists in the modern world. The frozen version is not alien biology. It is an old enemy with a proven method of survival that makes it different from some giant viruses found in permafrost. Pithovirus and Mollivirus are terrifying because they prove survival across deep time. But they infect amoebae. Anthrax is terrifying because it has already crossed from thawed remains into living animals and humans. It has already shown the root frozen carcass to soil, soil to herd, herd to people. The chain is simple enough to happen again. The question is how old the next release could be. The 2016 outbreak was linked to remains roughly decades old, not tens of thousands of years old. But anthrax spores can survive for extraordinary spans of time, especially in cold, protected conditions. Older animal burial grounds may hold strains that have not circulated in modern populations for generations. Some could differ from familiar strains. Some could appear in remote places where medical response is slow. Modern medicine can treat anthrax if it is detected early. Vaccines exist for certain high-risk groups and animals, but treatment depends on recognition, speed, access, and containment. In remote Arctic regions, those conditions are not guaranteed. A thawing carcass does not send a warning before it releases spores. Animals graze before scientists arrive. People handle meat, hides, bones, and soil before the outbreak has a name. That is why anthrax belongs among the prehistoric plagues scientists fear could return. It has already returned once from frozen ground, and it did not need imagination, speculation, or cinematic exaggeration. It needed only heat. The permafrost did not create a new disease. It preserved an old one long enough for climate change to hand it back to the living. The most unsettling lesson of the Yamal outbreak is its simplicity. No ancient temple was opened, no forbidden sample was mishandled, no researcher made a mistake. The ground warmed, a carcass thawed. Spores woke up, and a disease from the past killed again. Anthrax is the warning that the ancient world does not always return as mystery. Sometimes it returns as a fever, a dead herd, and a child who should never have met a pathogen buried before he was born. Number 5. Smallpox. The eradicated disease that may still be in the ice in 1980, humanity celebrated one of the greatest victories in medical history. The World Health Organization declared smallpox eradicated, a disease that had killed hundreds of millions of people, scarred survivors, destroyed communities, and shaped empires was finally gone from the natural world. The virus that caused it, variola, was no longer circulating among humans. Officially, it existed only in a few high-security laboratories. For the first time, a human disease had been deliberately erased, but that victory may have belonged only to the surface of the earth. Smallpox did not vanish because every trace of the virus disappeared from the planet. It vanished because transmission was stopped. Vaccination broke the chain from person to person. Outbreaks ended. Routine vaccination eventually stopped in most countries because the disease no longer existed in living circulation. But the dead were never part of that calculation in the same way. Across cold regions of the world, smallpox victims were buried in frozen soil, sometimes during epidemics that struck centuries before modern medicine. Some of those bodies remained sealed in permafrost. That is where the unsettling evidence begins. In Siberia, researchers studying old graves from smallpox epidemics found human remains preserved by cold. Some bodies still showed signs consistent with the disease. In certain cases, scientists detected fragments of variola virus DNA. They did not recover a fully infectious smallpox virus from those remains. That is important. But the discovery proved something the eradication story had not fully reckoned with. Pieces of smallpox can survive in frozen bodies for centuries. This changes the meaning of eradication above ground. In living human populations, smallpox was defeated. Below ground, in frozen graves, pieces of the enemy may still exist. They are not spreading. They are not causing outbreaks. They may be too damaged to ever infect anyone naturally, but they are still there, preserved by the same cold that protects skin, clothing, tissue, and bone. The permafrost does not care that the world declared the disease gone. What makes this frightening is not only the virus itself, but the world it would return to. Most people alive today have never been vaccinated against smallpox. Routine vaccination ended after eradication, and immunity in older vaccinated populations can weaken over time. That means, if smallpox ever did return in infectious form, it would not meet the world that defeated it. It would meet a world that has forgotten it biologically. Smallpox was not a mild disease. Historically, it killed a significant portion of unvaccinated people who caught it, and many survivors were left scarred or blinded. Entire societies once lived in fear of it. The modern world has medical systems, emergency vaccine reserves, antivirals, and surveillance networks. But those systems depend on recognizing the threat quickly. A disease believed to be gone can be slow to enter suspicion. The deeper fear comes from technology. Even if ancient frozen remains contain only fragments, modern science can now read genetic fragments, assemble genomes, and study extinct pathogens in ways previous generations could not. That knowledge can help preparedness. But it also means frozen graves are not just burial places. They are biological archives. Inside them may be instructions from diseases that no longer circulate, preserved in pieces by ice and time. This is why smallpox is different from the giant viruses found in permafrost. Pithovirus and Pandoravirus shock us because they are ancient and strange. Smallpox shocks us because we know exactly what it can do. It is not an unknown monster. It is a named killer from human history, one we defeated only after a global campaign of extraordinary scale. If it remains anywhere outside laboratories, even as damaged fragments, then eradication becomes more fragile than it sounds. As the Arctic warms, old burial sites are becoming more vulnerable. Riverbanks erode, ground collapses, cemeteries shift. Coffins and bodies once locked in frozen earth can be exposed to air, water, animals, and people. Most exposures will not release viable smallpox, but the possibility is enough to make scientists cautious, because with a virus like variola, the margin for error is very small. That is why smallpox belongs among the prehistoric plagues scientists fear could return. It may not be prehistoric in age, but it belongs to the same frozen danger, a disease preserved beneath the cold, waiting outside the living memory of modern immunity. Humanity announced smallpox dead in 1980. The surface world listened, the frozen ground did not. And somewhere in the thawing Arctic, old epidemic graves still hold the scars of a virus the world cannot afford to meet again. Number 4. Ancient Plague. The Black Death before history had a name. When most people hear the word plague, they think of medieval Europe, blackened bodies, mass graves, abandoned villages, and a disease that tore through the 14th century with impossible speed. But the Black Death was not the beginning of plague. It was only one chapter in a much older story. Genetic studies of ancient human remains have shown that Yersinia pestis, the bacterium behind plague, was infecting people thousands of years before the medieval pandemic, before classical Greece, before Rome, before the pyramids, and before writing had fully entered the world. The evidence came from bones and teeth. In ancient skeletons from Bronze Age Eurasia, scientists recovered fragments of bacterial DNA preserved inside human remains. What they found was not modern plague, and not exactly the medieval strain that later devastated Europe. These were older lineages, ancestral branches of Yersinia pestis, that had circulated across vast regions of the ancient world. Some of them were more than 5,000 years old. That means plague was moving through human populations when civilization itself was still forming. This changes the way we understand the disease. Plague was not simply a medieval disaster. It was a prehistoric companion of human movement, trade, migration, settlement, and animal contact. As people crossed steppe landscapes, lived near rodents, stored grain, traveled in groups, and created larger communities, bacteria found new ways to move with them. Long before the word pandemic existed, plague may already have shaped populations, migrations, and the collapse or transformation of communities we can barely reconstruct. But here is where the warning begins. Ancient plague lineages were not identical to the ones modern medicine knows best. Over time, Yersinia pestis evolved new traits, including adaptations linked to flea transmission and different modes of spread. Some Bronze Age strains may have behaved differently from later bubonic plague. Their surface proteins, virulence factors, and transmission pathways may not have matched the medieval version perfectly. In other words, the plague family is not one enemy. It is a changing lineage of enemies. That matters because modern immunity and modern surveillance are shaped by known strains. Doctors can treat plague today if it is recognized early. Antibiotics work against modern Yersinia pestis in many cases. Public health systems know where plague reservoirs exist and how it usually behaves. But an ancient lineage, preserved in frozen remains or old animal reservoirs, could carry biological differences that modern systems do not immediately recognize. It might not behave exactly like the plague we expect. The permafrost connection makes the fear darker. Plague has lived not only in humans, but in rodents, fleas, and burrowing animals. In cold regions, infected animals and humans could be buried in frozen soil, with bacterial DNA preserved in teeth, bone, tissue, or carcasses. Most ancient plague bacteria are dead, but the discovery of viable spores, revived microbes, and ancient organisms in frozen environments forces a difficult question. What if some bacterial lineages are not only preserved genetically, but biologically? Yersinia pestis itself does not form spores like anthrax, so it is not built for survival in exactly the same way. That makes a natural revival less straightforward. But frozen conditions can preserve biological material far longer than warm soil, and the deeper point remains. Ancient disease reservoirs exist in the ground. We have already found the genetic ghosts of plague in people who died before civilization became recognizable. The frozen world may hold more than ghosts. The most unsettling part is the age of the relationship. Humans have never lived completely outside the shadow of plague. It was there before history. It was there before medieval cities. It may have moved with Bronze Age migrations and infected communities whose names were never recorded. The Black Death was not a sudden monster appearing from nowhere. It was the return of something very old in a form that had learned new ways to kill. That is why ancient Yersinia pestis belongs among the prehistoric plagues scientists fear could return. It shows that some of humanity's most famous diseases have roots far deeper than our history books. The danger is not only that the medieval plague could come back, it is that the plague family contains ancient branches we barely understand. Branches that once infected humans and then disappeared from living memory. The skeletons tell us plague was there. The teeth preserve its code. The ground holds the remains of hosts that carried it. And as the frozen past opens, we face a darker possibility. The next plague may not be new. It may be an old lineage returning to a world that has forgotten how it first learned to fear it. Number 3. Methuselah microbes. The bacteria that outlived the dinosaurs in the year 2000. Scientists reported a discovery that sounded less like microbiology and more like something from a geological nightmare. Deep underground in New Mexico, inside ancient salt crystals, researchers found bacterial spores sealed away in a deposit believed to be around 250 million years old. That number is almost impossible to feel. It reaches back before the first dinosaurs rose, before mammals became dominant, before birds, before flowers, before every human ancestor anyone would recognize. Then the spores were placed in growth medium. They began to divide. The bacteria were not fossils. They were not only chemical traces. They were living cells in suspended form, capable of returning to biological activity after a span of time longer than the entire age of dinosaurs. The strain became famous as one of the so-called Methuselah microbes, named after the biblical figure associated with extreme age. But even that name feels too small. This was not old life. This was life that had waited through the rise and fall of entire worlds. The key to this survival is the spore. Some bacteria, when conditions become hostile, can form incredibly durable dormant structures. A spore is not ordinary sleep. It is a biological lockdown. Metabolism slows to nearly nothing. The cell protects its genetic material. It wraps itself in layers built to resist heat, cold, radiation, dryness, pressure, and chemical damage. In this state, a bacterium can stop participating in time in the way larger organisms do. It does not live normally, it endures. That is what makes the Methuselah microbes so unsettling. They showed that dormancy may not be measured only in years, centuries, or even millennia. Under the right conditions, it may stretch across geological time. A sealed salt crystal can become a time capsule. A frozen layer can become a vault. A deep aquifer, an ice core, a buried sediment, or a permafrost pocket can hold life in forms that modern biology has barely begun to catalogue. The reassuring detail is that the revived strain was not shown to be a human pathogen. It did not emerge as a plague. It did not kill laboratory workers. But the mechanism that allowed it to survive is not unique to harmless bacteria. Some of the most dangerous microbes in the human world also use spore, like survival strategies. Anthrax forms spores, tetanus forms spores, botulism forms spores. The same general logic that lets harmless microbes wait in ancient environments can also let pathogens wait in soil, carcasses, graves, and frozen ground. This is where the fear shifts from one organism to an entire category. If one bacterium can remain viable for extraordinary spans of time, then ancient environments may contain living microbial lineages that have not interacted with the surface world for ages. Some may be harmless, some may be beneficial, some may be scientifically priceless, but others may be unknown threats. The problem is that we do not know which is which until they are revived, cultured, sequenced, or accidentally released. Deep time also changes the question of recognition. A bacterium sealed away for millions of years may belong to a lineage that diverged long before modern medical databases existed. Its genes may not match familiar pathogens. Its metabolism may behave strangely. Its resistance profile may be unexpected. Modern medicine is powerful, but it is strongest against enemies it has already studied. The deeper the organism comes from, the less familiar the biological map may become. This does not mean ancient salt crystals or permafrost layers are guaranteed to release deadly plagues. Most revived microbes will likely be harmless or unable to survive in modern conditions. But biology does not need every possibility to succeed. It only needs one organism with the right survival tools, the right environment, and the right host contact. Methuselah microbes prove that the waiting period can be far longer than human civilization itself. That is why Methuselah microbes belong among the prehistoric plagues scientists fear could return. They are not frightening because one specific revived strain has already threatened humanity. They are frightening because they expand the scale of what survival means. They show that life can hide beyond history, beyond prehistory, beyond the entire age of familiar animals, and still retain the ability to wake. The most unsettling lesson is that Earth is full of sealed rooms we have not opened. Salt deposits, ancient ice, deep sediments, frozen soils, caves, aquifers, and buried organic layers may all contain microbial worlds disconnected from the present. Some have waited longer than humanity has existed. Some may have waited longer than dinosaurs ruled the planet. And if they wake, they will not be returning from history. They will be returning from a time before history had any possible witness. Number 2. Pleistocene parasites. The worms that moved after 42,000 years in 2018, scientists announced a discovery that made the frozen past feel alive in a way viruses and bacteria never could. From Siberian permafrost, researchers recovered microscopic roundworms known as nematodes. These tiny animals had been sealed in frozen ground for tens of thousands of years, dating back to the Pleistocene world of mammoths, woolly rhinoceroses, cave lions, and Ice Age tundra. They should have been dead. Their cells should have been destroyed by ice damage. Their bodies should have been biological ruins. Then the samples were thawed. The worms began to move. They did not merely contain ancient DNA. They were alive enough to resume function. They fed. They moved through the laboratory medium. They showed that a complex multicellular organism with muscles, nerves, digestive organs, and reproductive systems could survive freezing across a span of time longer than recorded history by many orders of magnitude. Until then, ancient revival stories often centered on viruses or bacterial spores. Nematodes changed the scale of the fear. That is what makes Pleistocene parasites so disturbing. A virus is minimal. A bacterial spore is built for dormancy, but a worm is an animal. It has tissues that can rupture, cells that can freeze, internal structures that should not easily survive thousands of years locked in ice. If nematodes can come back from permafrost after tens of thousands of years, then the frozen ground is not only preserving microbial fragments, it may be preserving complex life in suspended form. The nematodes revived in the study were not human parasites, ascites. They were soil-dwelling organisms that detail matters. They were not released as a plague, and they did not infect people. But the principle is what frightens scientists. Parasitic nematodes are biologically related to harmless soil forms. Roundworms, hookworms, whipworms, and many other parasitic worms use survival strategies that allow eggs or larvae to endure harsh environments. If harmless nematodes can survive deep freezing, then ancient parasitic forms may also have a chance under the right conditions. This is where the Ice Age carcasses become dangerous. Mammoths, woolly rhinos, ancient horses, bison, cave bears, and other Pleistocene animals did not die sterile. Their intestines carried worms, their tissues carried larvae, their dung carried eggs, their bodies were ecosystems. When they froze, parasites froze with them. For decades, those parasites were treated as paleontological evidence, dead traces of ancient disease, useful for studying extinct ecosystems, but not biologically active. The revived nematodes make that assumption less comfortable. The path back into the modern world would not need to be complicated. A thawing carcass emerges from permafrost. Its tissues rot into soil and meltwater. Eggs or larvae enter mud, streams, grazing land, or river systems. Animals drink from contaminated water or graze on exposed ground. Livestock become hosts. Wild animals become carriers. Humans enter the chain through food, water, handling carcasses, or contact with contaminated environments. A parasite does not need a dramatic airborne outbreak. It can move quietly through soil and bodies. Modern immune systems may not recognize Pleistocene parasites well. Humans and livestock today are exposed to modern worm species, and medicine has treatments for many of them. But ancient lineages could differ in behavior, host preference, life cycle, drug vulnerability, or immune evasion. They may fail completely in the modern world, or they may adapt to hosts close enough to the extinct animals they once infected. Biology often works through opportunity, not permission. The greater danger is not one worm crawling out of the ice. It is the proof that the ice can preserve animals, not just microbes. If tiny multicellular organisms can survive 42,000 years, then the boundary between fossil and dormant life becomes harder to define. The permafrost may contain eggs, larvae, cysts, spores, and resting stages from species science has never tested for revival. That is why Pleistocene parasites belong among the prehistoric plagues scientists fear could return. They show that the frozen past can preserve more than genetic memory. It can preserve motion. It can preserve hunger. It can preserve life cycles that were paused before civilization began. The worms that moved in the lab were harmless, but they carried a message from the Ice Age. Not everything in the permafrost is dead. Some things are only waiting for the thaw to finish. Number 1. Unknown megafauna pathogens. What the Ice Age is still carrying in the Arctic, the dead do not always disappear. Sometimes they return almost whole. A woolly mammoth emerges from a riverbank with hair still attached. A baby woolly rhinoceros comes out of frozen mud with organs still recognizable. Ancient horses, bison, cave lion cubs, and other Ice Age animals appear from permafrost, as if the ground had been holding them just beneath the surface, waiting for the right summer to let them go. To paleontologists, these discoveries are priceless windows into the Pleistocene. But biologically, they are something more disturbing. They are not just fossils, they are frozen ecosystems. Every living animal carries a world inside it. Its gut contains bacteria, its skin carries microbes, its blood may contain viruses, its organs may hold dormant infections, its intestines may contain parasites, eggs, larvae, and worms. Its tissues may preserve pathogens that were circulating through its herd, its environment, and the predators or scavengers that shared its world. When a mammoth or woolly rhino died and froze quickly enough, the cold did not only preserve the animal, it preserved parts of the invisible world that lived with it. That is what makes megafauna pathogens the most frightening entry on this list. Pithovirus, Mollivirus, and Pandoravirus were specific discoveries. Anthrax was a known outbreak. The 1918 influenza was a known pandemic. But frozen megafauna carry the unknown category, the viruses, bacteria, fungi, and parasites that belong to extinct animals, vanished ecosystems, and ancient disease network science has not fully mapped. We cannot name most of them because we have not seen them yet. The host problem is especially unsettling. Mammoths are gone. Woolly rhinoceroses are gone. Cave bears, cave lions, giant ground sloths, and many other Ice Age mammals are gone. A pathogen adapted only to one extinct host might fail in the modern world. But pathogens do not always obey neat boundaries. Many major human diseases began in animals. Influenza, HIV, coronaviruses, and countless other infections show that species barriers can break when biology finds an opportunity. A pathogen from an extinct mammal would not need its original host if it could recognize something close enough. Modern animals may provide that chance. Elephants are relatives of mammoths. Rhinos still exist. Horses, bison, reindeer, wolves, bears, and humans share broad mammalian biology with Ice Age animals. The match does not have to be perfect for a pathogen to experiment. It only has to enter a body, survive long enough to replicate, and find a route onward. Most ancient pathogens would probably fail, but in disease emergence, most failures do not matter. One success is enough. The route from thawed carcass to modern life is not imaginary. Permafrost thaw exposes tissue. Scavengers bite into it. Meltwater carries microbes into soil and rivers. Insects contact decaying remains. Humans handle bones, tusks, hides, and carcasses. Researchers cut into preserved bodies to study them. Local communities encounter remains as the landscape changes. Every exposure is a small experiment between the present and a biological past that has not been tested in thousands of years. The medical danger is recognition. If an unknown ancient pathogen infected a modern host, it would not arrive wearing a label that says Ice Age. It might look like a fever, a respiratory illness, a skin infection, a livestock disease, a neurological syndrome, or a cluster of unexplained deaths. Surveillance systems search for patterns they know. An organism from a vanished ecosystem may not match those patterns quickly. It would be called new, even if it was ancient. This is why the thawing of megafauna is different from ordinary fossil discovery. A bone is history. A tooth is history. But a preserved carcass with tissue, organs, gut contents, and parasites is also biological material. It may contain dead evidence. It may contain harmless microbes. It may contain valuable scientific data, or it may contain something still viable enough to test the modern world. That is why unknown megafauna pathogens belong at number 1. Among the prehistoric plague scientists fear could return. They represent everything we cannot list, cannot vaccinate against, cannot test for in advance, and cannot predict with confidence. The danger is not one named disease. It is the unexamined inventory of the Ice Age itself. The mammoth rising from the mud is not only a symbol of extinction. It is a reminder that extinction does not erase every passenger a body carried. The animal died, the ecosystem died. But under the ice, some parts of that world may have remained suspended, waiting without purpose, without memory, without malice. And as the permafrost opens, the final question is not what the megafauna were. It is what they are still carrying.